that's based out in Keladaba. Is that right? And then it became an official working group of the Endangered Wildlife Trust. Okay, we can frozen. Don't worry, back. Sorry. In 2005, the Department of Science and Technology recognized the collection as a national asset, and then we were transferred to the National Zoological Gardens of the National Research Foundation. So we were part of the NRF for quite a period of time. Wait. In 2012, we moved the entire facility from Nexa to the National Zoological Gardens in Pretoria onto the campus there into a dedicated building. In 2017, we had the most horrific revamp. We received funding for the replacement of the ceiling and the roof. It should have taken 6 weeks of moving out, or 6 weeks that we were moved out for. It took 11 months. So in that period of time, we needed to replace the roof, the ceiling, the floors, the walls, pretty much all of the furniture, because everything got so hectically damaged. In 2020, the National Zoological Gardens in its entirety was then moved to SANBI. So we've been one of the facilities in SANBI since 1st of April 2020, and it was moved as a going concern. And then also in 2020, we moved from research to collections management. So it's such a better fit now. We the same under the collections management, we fall under the chief directors of foundational biodiversity science, and there's 5 collections. There's the 3 herbaria, and then there's the SANBI Wildlife Biobank, and recently the SANBI Plant Biobank, which is now also based at the herbarium in Pretoria. So established around about 1994, we have about 250,000 samples, biomaterials, preparations for specified language, from about 95,000 individual animals, representing 250 families, 800 genera, and it's about 1,200 species in the collection. According to the threatened species represented in the Wildlife Biobank, according to the IUCN Red List, we've got 75 vulnerable, 47 near threatened, 37 endangered, 18 critically endangered, 1 completely extinct in the wild, and 1 extinct. So extinct in the wild is the northern white rhino, where there's only 2 species left in the world, and extinct is the western black rhino. We've got samples from 46 different countries around the world. This isn't field trips that we've necessarily been on, but we also rescue orphan collections. So these are deposits of collections that are put into the biobank. So the biomaterial types is part of the collection. So although we were part of the NRF since 2005, I've never seen SAIAB in my life. I've been to Grahamstown once and I never came here. So seeing this collection was a little bit eye-opening. We don't collect the voucher specimen. We don't have space for it. So ChatGPT helps you, or AI helps you with an elephant and a rhino in a building. We don't have space for that because that's ideally what we're collecting. So we rarely store the voucher. We rely on e-vouchers, which is your photograph, and then those are all appended to the database anyway, and it adds so much value to your actual sample as well, because you've got a collecting event, you've got your photographs, you've got all of your metadata, and you've got your sample. And then it's usually done as a chain of custody process. We were part of the Google Barcode of Wildlife project from 2013 onwards. I know SAIAB was also part of it. And by implementing this chain of custody approach with taking photographs of you, collecting the samples as well, all of your metadata, you can now 99.9% say this is a cheetah, because it's now been genetically verified, and with all of your photographs, you can show them that collecting event was a cheetah. So we store various biomaterials. It's not just in ethanol. We actually store blood spots, all from capillary tubes, also collected in EDTA top tubes and the red top tubes. And then these are stored as either on Whatman FTA paper or blood derivatives. And then also more recently, blood and ethanol. So they do almost a 1-to-1 ratio. It's the same as a buffer. And these go either to room temperature or -80 degrees. There's also tissue samples, either biopsies or notches, organs from post-mortem samples, and then also carcasses. So we're based at the zoo in Pretoria. So we've got an entire captive collection on our doorstep. So part of the biobank processing as well is for our team to go out with the vets for every routine operation, health checks, anything. We collect samples every single time the vets go out to work on any of the captive collection. And then also, of course, when they die, then we also get samples from postmortems. So these are stored at either 85 or in 85% ethanol, either on salt or silica, frozen at -20°C, or a 10% buffered formalin solution. Hair and feathers, just stored in envelopes or Ziploc bags, and these are at room temperature. Histopathology slides, so those are the formalin-fixed paraffin embedded wax blocks, and those are also stored at room temperature, but it has to be below 24 degrees, otherwise they melt. DNA, we store extracted DNA. If we give a sample out of a sensitive or an important species, we do ask for DNA extracts back when we know the lab technician almost. We don't want to do it for every student to say we have to have extracts back because you don't know how the sample is being treated. So we need to know that it's a good quality extraction, then we'll bank it at -80. We also have cell cultures. These are stored as fibroblasts, so it's a renewable cell line, and these are at -150 or -196°C. And these are collected and stored in quarter cc straws under liquid nitrogen temperatures. The SANBI Biobank workflow. The samples are collected, or it's an orphan collection that comes into the biobank. So before we catalog anything in the collection, we make sure that the data is there and the sample is there, then it's allocated a catalog number. Even by receiving orphan collections from universities or stakeholders, we match the sample to the data, then allocate a catalog number. That's why we have a backlog in one of our DNA banks. All collections have got backlogs. I was told otherwise years ago, and I got into trouble for it, but I argued the point, so I won. You will always have a backlog. You're always sorting out samples and making sure that you can match your sample to your data before you catalog it. Otherwise, you're making mistakes. Very important to know is that we use Specify. We have been using it. It started being implemented in 2012. It was fully implemented in 2014. So we've been using the database since then. I'm a big advocate for it as well, as Willem knows. I will fight people on it. A lot of people will fight about LIMS systems. LIMS cost money. Your software costs money. You need to pay your annual fees for it. Specify is basically free and it can do the same thing. The samples are then processed once they are in the biobank, moved into the various banks. So it's either room temperature, a DNA bank with all of our ultra low freezers, or into cryo collections. And the sample use is just, it's for so many different studies that you can use it. And the catalog into the collection. Currently, this is what our building looks like. We've got the various labs. And then also the banks. We've got a tissue bank, 2 DNA banks, the cryo bank in the top right corner, walk-in fridge and freezer. We've got a pathology bank outside for formalin samples because of the carcinogenic property, so they are kept outside. And more recently, we've had another walk-in freezer constructed outside. So this is a sample reception. I'll just go a photographic example of the biobank or a walkthrough. This is sample reception. So this is considered a dirty area in our collection. This is where everything gets processed. We are DALRAD compliant as a biobank as well. We had to be audited, and you have every 2 years, you have your compliance checked again. So one of the requirements here is that they actually want a BSL2 for all of our staff to work in, just because of the zoonotic nature of some of the potential tissues that you're working with. So we've got money through the Biodiversity Biobank South Africa project to actually buy a biosafety that can fit 2 or 3 people in it. So that should be procured by the end of the year. The room temperature tissue bank, you can see we're running out of space quite hectically. So this is where all of the samples on the left-hand side here, this is DMSO. We've stopped storing in DMSO because it's okay for normal genetic level studies, but for genomics and stuff like that, it doesn't work. Your extracted DNA is very poor. There's the Google Barcode of Wildlife project at the back there, where we've got the opened evidence bag, and then we've still got a reference sample that is sealed in an evidence bag. Those were supposed to go to the forensic science laboratory at the police, but they've stayed with us and they're happy for them to stay with us because we're a fully access controlled facility as well. And then all of the tubs with the red label on the front, those are all of the FFPE blocks. And then the front are the slides with the cut pieces of the FFPE wax. Using the microtome, you make your little slides for your histopathology. This is DNA bank 2, so we've got 4 degrees, -20 and -80. When we're sorting samples, they can go into various temperatures in this bank. We know that this bank is mainly for the sorting of samples. We have the second, is there a pointer? Is there a point on here? Can't see anything, my brain. Sorry. Thank you. This thermo freezer here, it's dedicated to the herpetology collection that we received from Prof. Crystal Tolley. So she was supposed to be audited as a lab or a biobank because she's got this vast collection of herp samples. But instead of managing access to the samples, she rather sent it to us. We now manage the collection for her. So we've dedicated a freezer just for those samples because it will be growing all the time as well. But it's in this bank at the moment because we're still sorting them. So we're matching the catalog number to the record, the sample to the record, and then we'll catalog it. So we've got a DNA bank 1. So we've got the -80 and -150 degrees Celsius running out of space with these as well. We're getting 2 new -80s before the end of the year. This is where all of the blood derivatives are stored. So we store in the 2 ml cryotubes. Everything is processed into the 2 ml cryotubes into an 81 well box and then stored in the various racks in the freezers. And then the extracted DNA is also stored. The cryobank, you can see that we're running out of space here as well. Everything is under liquid nitrogen. You can either store in a vapor phase or in a liquid phase. We store in liquid phase because we've got semen samples and fibroblasts, and they have to be maintained at the fibroblasts -150 or below, and semen cells -196, because it's 7 seconds and your semen store is thawed. So everything is below liquid nitrogen. This is probably one of the biggest costs of our operational budget, is the procurement of liquid nitrogen. But also through the BBSA, we're ordering a liquid nitrogen generator. So we'll have a little unit on site that can generate its own liquid nitrogen. We have a cell culture laboratory where we do the basic growing of the fibroblasts. So you started in a little petri dish, which is a tiny little piece of skin. Started in a petri dish, and as the cells grow, you passage them into bigger flasks, and eventually you freeze them. And also what's nice and exciting is we now have a karyotyping system. So we can look at chromosomes of various species. And just to know as well, or to note for all of you, is that we are a government facility. We serve as a platform to the bigger community. So if anybody ever wants to do something like chromosomes on fish, you don't have to go and buy your own, not as in fish chromosomes, but the actual species of fish, because I know that you get the fish banding or type of software, or when you're looking at chromosomes, you can come and use this platform. You can use our lab for growing of cells to see whether your protocols work. It is a platform and it is available to the bigger community. And then, of course, we've got a temperature monitoring alarm system. So we've got probes in all of our fridges and freezers and in our DNA banks, and it monitors the temperature with a half-hour delay. And often what actually happens is we notify the civil department to say, by the way, you had power failure, because we noted immediately. We don't have load shedding. We're very lucky we're on the same block as the Union buildings and also the Steve Biko Academic Hospital. So we don't have load shedding. If there is a power failure at all, we immediately turn over to generator. But because the biobank uses such a huge amount of electricity, we're also looking into solar. So the functions of the biobank, we provide adequate information and physical infrastructure to properly curate animal biomaterials for the benefit of the research community. This includes optimal temperature conditions for banking from room temperature down to -196, to secure and manage components and systems to govern biodiversity samples with benefit sharing and sustainable use of our heritage, species-specific sampling protocols for reference databases for legal trade, and for biodiversity management plans, so that's cheetah, leopard, lions, cranes, zebras, sun gazers, pangolins, and then also chain of custody sample collections for reference databases for illegal trade, which is the Barcode of Wildlife Project and also GEF7. And then to be the ideal platform in support of and advising to the custodianship for biomaterial collections. The participation and collaborations projects for others. So we are part of the African Biogenome Project, where we are giving samples through, which is an affiliated project of the Earth Biogenome Project and the collaborating project of the Vertebrate Genomes Project. The bottom left here is GEF7, so Barcode of Life. It all goes onto a database, your sequences or your barcodes for your legal and illegal reference databases. Chain of custody sampling protocols for illegal trade. We've helped with the EMIs and the National Prosecuting Authority. We're busy with the National Biodiversity Assessment 2025, where they're actually having a look at biodiversity collections in South Africa and where the gaps are actually sitting. So because BBSA is now hosted by SANBI, we're also participating in the NBA. Diplomics, I know that you're all quite aware of diplomics and submitting of samples. So my role in diplomics, or the biobank's role, is actually helping with the permits and the Section 20 compliance. But then the scientists in SANBI are also submitting samples. CMAPS, which has just come to an end now, which is the invertebrates collected just off the shore of Southern Africa. I think you were also participating in some of it. And then, of course, biodiversity biobanks. And then two committees, for IUC and Species Survival Commission. It's Animal Biobanking for Conservation Specialist Group, and then also the Pangolin Specialist Group. So the role of modern day biodiversity biobanks. So the OECD recognized the importance of having a wide network of biological resource centers for the storage and sharing of biomaterials, for the advancement of biotechnology, human health and research, and development in the life sciences. So molecular level investigations are being used in almost every field of biology to explore key questions about living systems and how they function. And the demand for novel foods, drugs, and other products is increasing. So the BBSA, or Biodiversity Biobank South Africa, it's important to ensure that South African species biodiversity is represented in national biobanks so that it can serve as a resource for research in a wide range of fields, including conservation of iconic and highly threatened species, crop and livestock stock improvements, and then also the development of new medicines or foods or industrial products. And the BBSA is one of the projects under the DSI's SAIAB project, or the roadmap. So years ago, a proposal was submitted to government about a biodiversity biobank kind of network. And the original proposal submitted was to actually have a single biobank. So do you centralize or not centralize? And that was a decision that government then made. They don't want to centralize. They don't want to make the community, the biodiversity community, submit all of their samples into one facility and it's managed by one top, person right at the top, that they have the say of everything. There, it's a facility to house all the multidisciplinary collections, so that'll be an absolute nightmare to manage. You'll need all of the expertise for the multiple collection curations. The budget would be huge because of the HR complement. You'll have to have so many specialists and scientists and technicians managing this collection that you would probably use your entire budget just on that. And also your facility. Your storage conditions are going to be so vast. I mean, if you think that you're storing bacteria, yeast, viral cultures, plants, foods, crops, everything, the expansion can never meet all of the users' needs. One person's going to get upset because theirs isn't expanded where somebody else's is. And then the terms of use, is it open or commercial? So then the BBSA, it's a distributed network of partners. So you keep your speciality in-house. So here it's multiple facilities to have dedicated collections. The expertise is for the particular collection. The budget can be evenly allocated. The storage conditions are applicable to your collection. So we need liquid nitrogen, so we'll make sure that we've got liquid nitrogen, for example. The expansion is determined by the user's needs, and then the terms of use can be decided upon per facility. And just the models of participation in the Biodiversity Biobanks network. So we get core biobanks and we get affiliate biobanks. Core biobanks are allowed to tap into the funding that is provided by government. And this is to make sure that your biobank is maintained at a really good level. And the biobanks that aren't quite at that level yet, they're given scope to expand and to better their biobank itself. So it serves national and international research and development. The expansion plan is based on the needs of the external user community. The access to the bulk of the samples. You actually want open access. You want people to make use of your collection. Otherwise, you're putting money into this collection and nobody's using it. So you want people to actually access your collection. You will have restricted samples, but these will be clearly defined. And then the benefits will mainly be at national level. And then you also contribute to development and agree to implement curation and data standards. So like we're doing with my visit to SAIAB, Session and I are talking about workflows. Willem's helping me with the next phase of the database. So it's a strong network that you can always tap into various people for improving. And then the affiliate biobanks, they serve in-house researchers, postgrads, and collaborators. The expansion plan is dependent on projects carried out internally. And then the access is to the bulk of the samples dependent on the individual ownership or the collector's permissions. And then the benefits are to the individual or to the institution. So it's my sample, I will get the benefit for it. That's what we've tried to move away from. And then the institutional level decisions on standards are then made. So some of the benefits of belonging to a national collection, access to funding to ensure expansion and growth of the collections and its holdings, absorbing orphan collections. So we were growing, but not all this time with valuable samples. You can only go on so many coals in South Africa and add those to your collection. We want samples with good metadata that have been collected for a research project that people just aren't using anymore. That's what we want to pull into our biobank. Appointment of staff to support expansion. We're a team of 3 full-time staff, and we're getting a new technician now. And then we also rely on internships. So support and troubleshooting of like-minded people. Seshni and I have kind of got the same roles in the collection. So it's nice if she can ask me a question, I can ask her a question. And the same with the other biobank curators or technical staff. It's a community that everybody can touch base with. And then also the reputation and reliability as a national collection. We aren't just a collection of SANBI. We are a national collection serving a purpose to the research and scientific community. The increased stakeholder contribution of samples. So also being part of SANBI, part of a national or being a national collection now. We've got some really good stakeholders that are contributing samples. We get a lot of our samples from rehab centers now, which is an indigenous wildlife. We weren't necessarily growing with indigenous wildlife. We were getting more from things like captive facilities, which is a lot of exotics, and missing out on all of the indigenous wildlife. A lot of the universities are now donating their sample collections that they're not using anymore, but also just maintaining their collections for them. They don't want to go through the whole DALRAD process of becoming a biobank or a collection or a lab. So the samples come to us and then the researchers access it that way. We're also busy talking with Ezemvelo KZN for batches of samples from them that they've got over years as well. That we will manage the collection on behalf of them. The people wanting use of the samples will still approach their project committee to use the samples, but we will manage the access to the samples, making sure all of the permits are in place, so whether it's TOPS, Section 20, all of the legal compliance. Another big thing with our biobank is our expansion project. So you could see that some of the banks were like quite tightly packed. We're getting funding through the Biodiversity Biobank for an additional probably 144 square meters. But if we get our top option, we'll go double story and put office space at the top. So it'll be another walk-in freezer, more office space, and that's why I put double story, a carrier typing lab and library, another room temperature tissue bank, and then another DNA bank for ultra-low freezers, and a DNA extraction lab for high-risk samples. Because we're getting a lot of samples from all over the country, sometimes there is a problem with Section 20 for the extraction. So if we've got a BSL2 level lab, they're quite comfortable with us extracting, not things like foot and mouth disease or anything at risk of damaging our entire captive collection as well, because we are based at a zoo. And then also assistance to the Biodiversity Biobank South Africa community. So we're an established collection providing assistance to small and newer collection. So this could be in terms of SOPs, staff training, technical and curation, setting up fields of use for collections database. I won't do the collections database, that I'll point them to Willem or Wesley. But we can show them the fields that are quite important to capture, and then also optimal sample collection and storage. Thank you to SAIAB for, well, not really for inviting me and making me do this, but thank you anyway. Thank you for Willem and Stephanie for your time as well. It's been good and it's lovely seeing the collection. And then of course to SANBI and to BBSA. Okay, thank you.
Thank you, Kim.
No, you did.
Any questions from the floor? Yes, Martin.
Thanks for that, Kim. How do you navigate the ethics space? Various ways your samples arrive at your doorstep.
So when samples arrive, because we're getting them from, we actually chatted about this earlier today. Willem said be warned. So we're getting it from tertiary institutions. So we hope most of the time there was ethical considerations before collecting the samples. They've got all of their permits. When they actually bring the samples to us, we have all of the copies of the permits, all of the data, what permits it reflects to. So we've got copies of all of that. There is also that pre-exemption period kind of thing. You know, there has to be a cutoff date where you didn't have Section 20 necessarily. We only implemented it in 2014. Even though it stood for how many years beforehand. We have to be mindful that there need to be permits in place. There has to be ethical considerations when absorbing those into our collection. And it can be a long answer or can be a short answer. We try and get as much as we can before accepting the samples, as much information, as many permits, Section 20s, permissions, even land use permissions when we absorb that collection. There will be a point where somebody doesn't have something, and then do we accept it or not? So even, I mean, we accept samples from hunters. Well, we did in the past, but they've already got their collecting permit or a hunting permit. So that already is a legal document. Everything has to have some kind of paperwork behind it before we accept it into the collection. Before we allow access to the collection, they have to do a request form. They have to show all of their permits before we release any samples as well. So we don't release anything without the necessary permits. But it's not that it's a gray area with accepting samples. There's a lot more levels to it. Because we're a national collection as well, we don't necessarily want to turn something away. There was a factor when people went to go and collect those samples, there was some kind of compliance in place. That's what we're hoping for anyway. Whether somebody's got something from 20 years ago that they went to go and sample in the field museum collect or something like that, they had no permits whatsoever. That's I think that'll have to be evaluated on a case-to-case basis as well. Does it answer it? Or is it very all over the place? Yeah. I think that was our discussion this morning, was also all over the place.
Are you guys accepting roadkills?
For pangolins, yes, because we're the central repository for South Africa. Otherwise, not really. It's illegal actually to pick up roadkill. So unless it's somebody like Endangered Wildlife Trust where they've got like a permit to collect roadkills, we're also in chats with EWT about samples coming to the biobank. But as it stands, no, we don't really accept roadkills.
So I see that you're using specified, but is the data published anywhere so that when anyone can access it? So.
No. So that was one of the, through GGBN, the Global Genome Biodiversity Network, and GBIF, we will make data sets available. But there we have to black out a lot of locality information. So we can maybe do it down to a sample level. This is what's available, and maybe provincial. It was collected in Northwest. But otherwise, we've got too many sensitive species that can actually be, even if it was a rhino collected in 2005, there was a record of a rhino being at that location in 2005. You don't necessarily want to open up that farm to people going and sticking their nose in there.
Any other questions? Yonela?
Do you sequence your samples for identification purposes?
mm,
So, and how are you dealing, are you just relying on the pictures submitted? If there are any misidentifications, how are you dealing with that?
so when we go and collect samples ourselves, it's a trained person to do chain of custody. We're trained by the police, the National Prosecuting Authority, and the EMIs, so we'll do the chain of custody ourselves, especially for things like Jeff7. You have to be a trained sampler to be able to do it, to take the samples. So when those samples come in, we 100% know that that is the sample with the picture. So that's like 100% accuracy. When we're getting these samples in from different other collections, like rescuing these orphan collections, we have to look at the researcher behind it. I mean, you can't always 100% know that somebody has submitted as a cheetah and it is a cheetah. We have to take them, we need to match the sample to the data. We can't always 100% accurately say, yes, it is a sample. We don't sequence it. There are projects for barcoding of different, so say we get a sample request for barcoding, even if it's internal for one of the students that they want to have a look at 10 cheetah sample in the biobank for barcoding, then they will send the results to us. But other than that, we can never be 100% sure because we don't have the voucher in a lot of cases. Even now, with samples coming into the biobank from universities, they never have photographs.
Okay, we're going to take one online question and then we'll go to Vanessa. So the online question on the topic of roadkill, will you accept roadkill, example, leopards collected by provincial conservation officials?
Yes. Yeah.
All right, and then Vanessa from the
I
floor.
was just going to ask, do you deal with things like strandings of whales and dolphins and anything like that, or is that sort of fall under roadkill type?
We don't deal with it ourselves, but we've got stakeholders that collect the samples, and there's a few of the SANBI scientists working on some of the like stranding species.
Okay.
Then we'll get the samples like that. Yeah. Yeah. What we've also done is the Jeff7 project with the legal and illegal reference databases, and the biobank. We're working very closely together. So a lot of our contacts are now part of the Jeff7 contacts as well. They're keeping carcasses for us, because then you can do your whole chain of custody sampling thing. You've got your animal in hand, you can take really good photographs of it, so you can verify that that is the sample. And also mostly it's indigenous wildlife species. We've got a few DFFE collaborators as well that will collect samples like from Marion Island or whatever, that will all come to the biobank.
And what happens to things like abalone? Does that fall under the Jeff7, the illegal?
No, that's, I don't know who does, do you do abalone?
We have abalone, yeah.
Oh, So we're still doing the birds, mammals and reptiles.
Okay.
And then it's, you're doing all of the marine and aquatic, all
Yeah.
aquatic, yeah.
Any other questions? All good, okay, cool. Thank you so much, Kim.
Thank you. Can I? I'm going to try it.